We use the scientific method every day. For example, let's say you lost your phone. That would be an observation that you make using one of your five senses. So you ask a question, where's my phone? Now, there might be some research that could help you answer that question based on the most common places to find it. But generally, we just skip to the hypothesis and we say, maybe I left it on the kitchen table. Maybe I left it in the car. We experiment by checking those places. Now, in a more scientific setting, observations can be quantitative, where we're making a measurement, like milliliters or grams, or qualitative, that has to do with like shape or texture or color, where there's not a number involved. When you're writing a hypothesis, try to show the relationship between the two variables. If caffeine is related to heart rate, what do you expect will happen? More caffeine, maybe higher heart rates. The experiment that you conduct then will have some variables. The independent variable is the thing that you determine, the thing that you change. For example, you change the amount of caffeine, or you determine how much time goes by. The dependent variable is the thing that you're measuring. You're measuring the heart rate. You might divide your experiment into two groups, where you have a control group that gets a placebo, something that looks like the change, but it's not, and an experimental group, a group that actually experiences some changes that you're trying to investigate. When you're thinking about a hypothesis, try to use these words. If one variable is related to the other variable, then what do you expect will happen? For example, if caffeine is related to heart rate, then I would expect the more caffeine, the faster the heart rate. Or if we look at light and photosynthesis, if light intensity is related to photosynthesis, then I expect that the greater the light intensity, the faster the rate of photosynthesis. But I might have to do some research. So photosynthesis is this process where plants take carbon dioxide and water, convert them into sugar and oxygen. The question is, what do I measure? See, plants are making sugar and they're making oxygen, but plants use sugar. So they're collecting carbon dioxide from the atmosphere, they're collecting water from their roots. And with that, in the help of some sunlight, they are making sugars that they use for bark and leaves and fruits, and they're also making oxygen. But oxygen is a waste product, so I'm going to measure the amount of oxygen that the plant makes. So I set up an experiment that looks something like this. The test tubes that are closer to the light would have more light intensity. We expect the rate of photosynthesis to increase, and it's going to show that rate by the amount of oxygen produced. So if you look at the little plant in the test tube closest to the light, it's producing oxygen at a very rapid rate, whereas the plant farthest away is producing oxygen at a much slower rate. That confirms my hypothesis. If I have multiple experiments that are testing different hypotheses, but they can all be connected or related, we might call that a theory. So for example, the cell theory combines several hypotheses. All living things are made of cells. That's one. Then a hypothesis like the cell is the basic unit of structure and function. That's another hypothesis. Then a third one that says something like all living things come from cells, or cells come from other living cells. A law is a more mathematical way to describe something in science, like the law of gravity or the second law of thermodynamics. Those are more mathematical. A fact is a true statement that we often use in science, and a principle is based on facts. So for example, we might say something like, healthy eating leads to a better quality of life. Well, that would be a principle based on the facts of nutrition. So that gives you a little overview of some of the things involved with the scientific method.